Hi everyone. So what we're going to try to do today is continue our discussion on multivariate methods. And so if you recall last time, we played around with principal components analysis, which is one of the more standard traditional methods for thinking about multivariate data. And we can remind ourselves that we were able to rotate either a correlation or a covariance matrix in such a way that we could explain some portion of the variation condensed onto particular axes. So we could keep track of how things covaried with one another, and we were able to understand what the important predictors or what the important components were for each of those axes, and basically understand what the structure of our complex data look like. And as I mentioned, PCA can be a really handy exploratory data analysis tool to try to understand patterns of variation within the data, and it can also be an effective way to ordinate or to show a picture of what might be interesting patterns in the data. And I also mentioned at the end that it's totally fine, or at least it's not wrong, if you were to do kind of traditional linear models on the scores for any given axis. So let's say you have PC axis 1, it explains 30% of the variation. You could save all of the PC scores for all of your observations on PC1, and you could do something like an ANOVA or a regression if you had other kind of predictors you wanted to put in there. And there's nothing inherently wrong with that approach. It becomes a little abstract because you have to remind yourself that you're doing statistical hypothesis testing on a particular principal component, and it's only describing a particular amount of the variation. So you have to kind of keep that in mind. The other potential problem with doing that is that principal components analysis is trying to maximize the amount of variation in the data on those axes. But it's not necessarily trying to find the important components of the variation that might help you discriminate between groups, or maybe the important components of the variation that might covary with some predictor that you would want to put in the model. So that is an important limitation to doing that approach. And so that brings us to thinking about, well, how do we do kind of formal hypothesis testing on multivariate data? And there's a long history of that. And if you dig into the literature, especially in the 80s and 90s, one commonly used method was called MANOVA, which is multivariate analysis of variance. And that was a common method that was used. It has somewhat, I think, fallen out of favor. I think it's been probably at least 10 or 20 years since I have seen a publication that uses MANOVA, at least in an ecological or an evolutionary context. I can't speak for other fields. And one of the reasons why it somewhat fell out of favor is that it was shown to be incredibly sensitive to deviations from multivariate normality. So just like when we are doing a straight up ANOVA, at the end of the day, we are assuming that all of our things are random variables drawn from some kind of a normal distribution. In particular, we're assuming that the error term is going to be distributed normally. And, we can kind of drink the Kool-Aid and be okay with if it's not quite normal, and we know that ANOVA models are robust to violation of the assumptions. It turns out that MANOVA wasn't as robust, and that could potentially lead to a lot of erroneous inferences when you do those types of analyses. So as a consequence, MANOVA has somewhat fallen out of favor, and it's been replaced with a bunch of different things, but in a general sense, it's been replaced by what is referred to as constrained ordination. And constrained ordination is just what it sounds like. It's an ordination, but you're going to constrain you're going to constrain the ordination to the bits of information that are actually relevant to your question. And so if you want to know how a big multivariate data set might covary with elevation, you would only really want to extract the interesting parts of the variation in that large matrix that actually have something to do with elevation. You wouldn't want to dump all of the information into the model, which is what we do with a PCA, right? And so these constrained ordinations are going to give us a means to do hypothesis testing on a multivariate data set. And there's a number of different flavors of these. I'm going to focus on one, which is called redundancy analysis. If you recall, we played around with that RDA function. And we would give it the matrix, and then we give it the little squiggle, and then we just said 1, and that's basically just only estimating the intercept, so that's going to be the exact same as a principal components analysis. Much like if you set up a linear model, like just a standard regression model, and you asked only for the intercept, it's just giving you the mean of the data. And so when you do what's called a redundancy analysis, and you do that little (1), that is basically just saying it's an intercept-only model, so it's going to end up being the exact same as a principal components analysis. And there are a few other types of constrained ordination that are commonly used in evolution and ecology. One that immediately pops to mind is what's called a canonical correspondence analysis. And it, in principle, is very similar to how an RDA works. The only difference is that a CCA, as it's called, is built more for situations where you have a unimodal relationship. So let's say that you did have a gradient of elevation and you had a bunch of things that you measured about soil chemistry or whatever it might be. A CCA would be more appropriate if the distribution of the important components of all of those different aspects of soil chemistry followed some kind of a unimodal type distribution, where maybe at the mid elevation it was that you had the largest values, and then as you get to the extremes you have less and less of particular compound, let's say. Whereas a redundancy analysis is originally built a little bit more, not a little bit more explicitly, for thinking about things that occur on a gradient. So, imagine a regression line, essentially. That would be something that occurs on a gradient where there's a little amount here, and then as you increase elevation, you have more of it up there. And so the structure of both of those are very, very similar. When redundancy analysis was originally presented to the peeps, as it were, it was really sold as a way to deal with continuous predictors, if you had a lot of continuous predictors that you might want to put into the model. But now we all know that there's no difference between a multiple regression and an ANOVA, because at the end of the day, all we have to do is dummy code categorical things, and voila, we have a multiple regression. So redundancy analysis will take anything that our standard linear models would take. So it would take continuous, discrete predictors and interactions between those two, and however you would want to set up your model. The only difference is that on the left side of the formula, you are dealing with a matrix as opposed to dealing with just a univariate vector. And so how does redundancy analysis work? Redundancy analysis, behind the scenes, what is really happening is the computer is actually doing a bunch of univariate regressions, or ANOVAs, however you want to think about it, on all of the data. So it's making a bunch of linear models for each column of data, for each unique thing in our multivariate data set. And from there, it's calculating what the expected values are. So if you want to think about this as like a traditional regression, those are where the points are that are on that regression line. And so that's what we mean by the constrained analysis, because what we're going to do then is we are going to ordinate the predicted values. From those predicted values, just like when we ordinated in just a typical principal components analysis, we're going to get an eigenvector and we're going to get eigenvalues. The eigenvalue is going to tell us how important an axis is, and the eigenvector is going to tell us how important each of those components are to that particular axis, right? We can think of those as loadings. And so we're going to make that model to deal with all of the predicted values. And then we're going to have the eigenvector, which is going to tell us the weightings. And then we're going to go ahead and challenge our observed data using those particular eigenvectors. And the consequence of that is going to be that we are only going to really be thinking about the important components of the variation that have anything to do with our particular, with whatever particular constraint we decided to put on the data, so the predictors in our model. And so when we run an RDA, we're going to have certain number of predictors in the model, and then we can say this amount of variation is explained by those predictors, and then we can think about looking at the different axes of our constrained ordination to interpret what are the important parts or predictors of our data that we're able to understand across, whatever the predictors are we wanted to put in the model. Right, so what I'm going to do is I'm going to jump on into R, and we can just slowly walk through how to interpret it. If you understand principal components analysis, I think that the redundancy analysis will come on pretty easily for you. So let's jump in. So I'm going to just go through the notes that were posted, or the code that was posted. And so the first thing I want to do is I just want to remind ourselves, what the heck we're doing. And so we'll go back and we'll play with the same butterfly wing data that we played with when we were introducing PCA. And so just to remind ourselves, I'm going to go ahead and do another PCA, or a good old-fashioned PCA, as I like to say. I'm going to say scale equals true, so that way I'm dealing with the correlation matrix. I don't have to deal with the fact that certain things might have a higher variance than others. I'll just standardize everything to 1. So we're on the nice little correlation matrix. I run that, and then just to remind ourselves what we got going on over here. Principal component 1 is explaining 40% of the variation. Principal component 2 is explaining 18. Principal component 3, 6.5, so on and so forth there. So we know how much each of our principal components is explaining. Then we can go down here and we can look at the eigenvector. And we can look at the relative importance of each of these measurements for PC1 and PC2 and so forth, right? And so here we just have our nice principal component analysis. And then if you recall last time, I made a pretty plot. Let's see how my pretty plot looks. And there's my pretty plot of my three different populations, and this is the ordination of principal component 1 and principal component 2 for these different populations of butterflies. And you recall, when we were thinking about just looking at the principal components analysis, just by looking at that, we can eyeball it and say, well, principal component 1 seems to be doing a pretty good job at least discriminating the green population from the blue and the red population. Principal component 2, it doesn't really look like there's a lot going on there because everything is just kind of lumped up here around 0 of PC2. So PC1 appears to be important. And again, it's totally fine to go in there and interpret that stuff and call it a day. But we might want to do explicitly some hypothesis testing. Like maybe we want to know if populations are actually significantly different from one another or not. And so basically what we're asking is, would we expect that pattern if the null hypothesis that all populations were the same was actually true? And so this is where we can use a redundancy analysis. And so here I'm going to look at the PC of the butt wings, but I'm going to do it truly as an RDA, which means I'm going to do it as a constrained analysis. And I'm going to set it up just like I would set up a linear model. I'm going to say that my matrix, my butterfly wing matrix, is some function here of pops. Okay, so again, behind the scenes, what's happening is the computer is doing basically a factor-based ANOVA, if you want to think about it that way. It's just a linear model where the predictors are populations. So it's going to come up with and estimate a slope and an intercept for that linear model, and then that's what we are going to use to come up with the eigenvectors, and then that is what we're then going to put our observed data into so that we can look at the ordination of the constrained data. And what we're going to get out of that then is, for our constrained axes, those are going to do their best to capture the important components of the variation that can be used to tell the difference between those populations, as opposed to just simply trying to capture all of the variation that's out there. So I'm going to go ahead and I'm going to do that. And there it did. I did it. And we can look at a summary of that object. just to kind of see how it differs maybe slightly from when we did it as a PCA. And so first, we again, we have the same inertia as we had before. But here we have now a component, a variance component, that is the constrained part of our ordination, and a variance component that is the unconstrained part, so all the leftovers. Essentially, it's the residuals from all of those linear models. And then we can also see what the proportion explained is. So here, I'm able to explain 32.17% of the variation in wing morphology by partitioning that variation among populations. So that might be kind of interesting. You can kind of interpret that as a global R squared, if you would like. We can also see the individual components, and notice here that we have RDA1 and we have RDA2. Well, we have 3 populations, and remember how dummy coding works. One of those populations is getting lumped into the intercept. So there's basically 2 slopes and an intercept associated with this model. So as a consequence, we have 2 constrained axes. We have RDA1 and RDA2. And then after that, they start being called PCs. And that is just the ordination done just as we did it with principal components analysis, except here we're doing the ordination on all the leftover variation after we've partialed out the effective population. So RDA1 and RDA2 are our constrained axes. They're about our predictors, and they should tell us something about what components of the variation are important for discriminating among populations, whereas the PC axes are basically just the residuals. They're actually not really all that terribly interesting. And so we have our 2 RDA axes. The first one is explaining 30% of the variation. The second one is explaining 1.9% of the variation. So clearly, RDA1 is much more important than RDA2. But now we can take a look and see what that plot looks like. And it will look slightly different. So here is our principal components analysis. This is our redundancy analysis. And in this case, they look kind of similar. And that's primarily just because the green populations are a lot different from the blue and the red. And it turns out that the variation that was captured by the PCA is also the important components of variation that can be used to discriminate among those populations. But they do look different. RDA plots tend to look nicer, if you will, because they tend to do a much better job of pulling things apart to draw your eye to them. And yeah, and so you can see that, you know, RDA1 is 30% of the variation, and that's clearly separating out those green from the red and the blues. And then RDA2 looks like maybe it was trying a little harder to pull apart the blue and the red because they're so similar to one another, but it still doesn't look like it did that kind of a bang up of a job at it. So the plots will look a little different because now here we are plotting the constrained axes. And so now what do we do with that? So right now we have a pretty picture, and we could leave it at that and walk away with that. Just like with a linear model, you can run a linear model, estimate parameters, and walk away from it. There's nobody telling you that you have to do null hypothesis testing on it. But perhaps we do want to use, or we do want to do some kind of a null hypothesis test on it, because that's oftentimes what the objective is. And so much like a normal ANOVA, we can look at the amount of variation explained by our model versus the amount of variation not explained by our model. So recall in an ANOVA framework, we are using the F statistic, which is the ratio, which is the fraction of the mean squared of our model divided by the error mean squared. And so the variance component that we can explain divided by the variance component that we cannot explain. From there, we calculate an F statistic, and once we have the F statistic, we can go to the cumulative distribution function for the F distribution with the appropriate degrees of freedom and ask ourselves, what is the probability that I would get an F value that extreme or more extreme if indeed the null hypothesis were true? And so that's great. We should understand that framework of how it works, and we can do the exact same thing in multivariate space. We can still think about the distance from our predicted values to our observed values. We can square that term and then divide it by the appropriate degrees of freedom to get a mean squared. The problem is that there is no closed form solution for what the null distribution should look like. In other words, there is no multivariate extension of an F distribution that we can use in an ANOVA framework. So what's the solution? The solution that we'll do is permutational tests. In other words, what we'll do is we will simulate the null. And the null is a statement of no effect, which means that we can take all of our data, we have all of our populations, and we have all of our data, and then we can just randomize what population, what row goes with what population, and then we could calculate an F statistic for that. And then we can do it again and calculate an F statistic for that. And then we can do that thousands and thousands of times so that we will have a null distribution of what we would expect the F distribution to look like if the null were true, because that's what we're simulating, is the null being true. And then we can take that null distribution of F values and compare it against our observed F statistic from our ANOVA. And so the important difference here is that we don't have a closed form solution like the F distribution. We have to simulate what we think that simulation or what that distribution should look like. And so technically, these are not F statistics that we're going to be using. They are what are referred to as pseudo Fs. They're fake Fs. They really want to be an F. They're calculated the same way, but they just don't behave like a true F because they're not based upon the F distribution being the hypothesized sampling distribution where those F statistics are coming from. And so yeah, so we're going to do permutational tests, very similar to basically what we did when we introduced comparing groups. And we had those few examples where we could play around with doing permutational testing to find out whether two groups were different from one another. We're able to do the same thing here. And so we do that using a built-in function technically called ANOVA.cca, but R is smart enough that if you just type in ANOVA, it will know what to do because it's an object-oriented language and it's smarter than we think it is sometimes. And so there's two things to think about when doing permutational testing. The first thing is you want to set your seed. Because again, computers don't truly generate random numbers, they generate pseudo-random numbers. And so we're going to want to set the seed. That way, every time we do this analysis, this same analysis, or if somebody else tried to recreate our analysis, they would get the exact same answer. The second thing that we want to pay attention to is the number of permutations. And the default for this function in R is 999 permutations, which is simply not probably going to be enough. You'd like to do a lot more than that. Here I've set it to 99,999 permutations. So I really feel as though I'm doing a pretty good job at exploring what that null distribution of Fs should look like. So this approach is what is called a permutational ANOVA or a PERMANOVA. And there's a caveat here. And some of you that deal with like molecular type data or have done these types of analysis before have probably seen PERMANOVA used. And so I put this in the notes that this type of hypothesis testing is a PERMANOVA, permutational ANOVA. But there's a little bit of confusion there because there is actually a method that was branded as PERMANOVA. And it was a method that was published as PERMANOVA in 2001 by Marty Anderson. And it turns out mathematically it is identical to a method that was published in 1992 by Escoffier, where Escoffier built it specifically to deal with genetic differentiation using sequence data, whereas Marty Anderson, when she published her version in 2001, she was thinking about community data, like, you know, birds in the forest or trees in the forest or microbes in a ditch, whatever you want to think about as the community data. The PERMANOVA method that would be associated with Anderson 2001 is similar to an RDA, except it's looking at Euclidean distances from a centroid as opposed to distances based upon expected values from multiple different linear models put to the data. They tend to give very similar answers, but they are, in their heart, under the hood, they are doing something a little differently. So I just wanted to mention that because what we are going to be doing here is a PERMANOVA, but we're doing it with a redundancy analysis as opposed to doing it with a PERMANOVA analysis. So that's just a little something to help confuse you, because I'm sure that, you know, why not? It's the end of the semester, time to get extra confused. But just be aware of that. You see that a lot. Right. And so because we're dealing with linear models, one thing behind the scenes in an RDA, we're dealing with linear models. One thing that we're going to probably want to keep attention to is whether or not the underlying assumptions of those models are being met. And an important assumption with these types of analysis is homogeneity of variances, right? We don't want certain groups to be really variable and other groups to not be very variable, because that violates one of the assumptions. And so this is something that oftentimes, virtually always, either people aren't aware of or they just conveniently ignore when they're doing redundancy analyses. But it's worth at least checking to make sure that you're not violating those assumptions too dramatically. And an easy way to do that is to use a built-in function in vegan. So I guess it's not built-in, it is a function in vegan called beta disper. And without getting into the weeds as to why it's called beta disper, basically what it is doing is it's doing a multivariate Levene's test. Recall that the Levene's test was a test that we could use to compare the variability of samples, where we take the distance, the absolute value of the distance from the median, and then we use those as response variables to compare the dispersion between two groups. This function does a multivariate analog to that. It's essentially looking at the absolute value of the distance from the multivariate centroid among groups, and then asking whether or not certain groups are more dispersed than other groups are dispersed. And so what beta disper requires is a distance matrix. And so let's remind ourselves really quickly that these are our data, right? We have all these different butterfly wing measurements in this big giant matrix. And what I can do is I can turn this into a distance, and I will do that by saying, let's take the Euclidean distance. And now what I've done is I have the Euclidean distance between all of those data points. Since we're interested in doing this on the correlation matrix, we should also probably look at the z-transformed. So I think I've already done this, but I'm just going to create another object. ZB wing is going to get BW ing. I want to apply it by columns, and I want to scale it. Okay, so now I've z-transformed all of our raw data, and I will go ahead and I'll take the Euclidean distance of that. So now I have Euclidean distance between all of those points. And when we're saying Euclidean distance here, you know, if it were a simple two-dimensional thing, you know what the Euclidean distance is. It's the distance between 2 points. And then I guess if it were a three-dimensional object, it would still be the distance between 2 points. And then it becomes a little abstract for us humans to think about higher dimensions. But here we're looking at the Euclidean distance between points in however many dimensions we have. I honestly don't remember. What do we have? What is dim? Yeah, so here we have a 24-dimensional space, and we're looking at the Euclidean distance between all the points in that 24-dimensional space. So now once we have that, what we can do, now that we understand what it is, I'm going to go ahead and put that in here to create a beta disper object. And let's see here. Oh, I don't want to. Oh yeah, I got to tell it Euclidean. There's my problem. Euclidean. All right. So now I have this beta disper object, and now I can simply do an ANOVA on that. And it looks like there is no significant differences in the dispersion between our various groups. So now we can go ahead and we can start to feel good about what we're going to do, which is going to do the hypothesis testing. So let's remind ourselves again what our model is. I will go ahead and I will cut and paste that just so that way it's right next to where we're doing the ANOVA, or the PERMANOVA, as it were. So here's our RDA again, right? This is what we ran to do our constrained ordination, and now we want to do the permutations. And so I'm going to take that object, I'm going to randomize all of the individuals, all of the rows, I'm going to calculate that F statistic, and I'm going to do that 99,999 times. And that's going to give me the null distribution for the pseudo F. And then I could ask, what's the probability that I would get my pseudo F that I got if the null hypothesis were true? So we'll go ahead and we'll set our seed so everyone has the exact same answer, and we'll run that ANOVA. And then it will take a couple of seconds because the computer's thinking. There it is. And so here we can see, here's our F statistic, right? Here's our degrees of freedom, but again, none of that matters because it's not a true F. And so our p-value that we got here is based upon the permutations. So the probability that we would get an F statistic of 12.096 or greater, given our data, conditional on the null hypothesis being true, is 1 times 10 to the negative fifth, which clearly passes our threshold of an alpha of 0.05. So we reject the null hypothesis. We'll accept the alternative that there is a difference among populations. And so once we see that there's a difference among populations, we might be interested in understanding which populations are different and why. And just as there is no closed solution for an F distribution, that means that we can't use our traditional Tukey. And so one solution to this is, once you have the green light to go ahead and do it based upon our ANOVA, which says that there is a significant difference among populations, is we can do a bunch of pairwise comparisons and treat it as a post hoc test. And so what I'm doing here is I'm going to say I'm going to look just at L versus C, right? Because if we remember our populations, those are our different populations that we're looking at, L, C, and M. So I'm going to restrict it now to just L versus C. So this is going to call out only the rows that are both either an L or a C. And I'm going to make a subset of the data for that purpose. And I'm going to do an RDA, just looking at the L versus C data, and then just comparing the populations that are L versus C. Otherwise, everything is the same. I'm going to go ahead and run an ANOVA, arbitrarily set a number seed. And there I go. I'm doing, once again, 99,999 permutations. And aha, still significant. Which means right now we feel pretty good that L versus C, L is different from C. So now, next we can try L versus M, which is the exact same code. The only thing different here is that I'm putting L and M instead of L versus C. So I'll go ahead and I'll run that, set an arbitrary number seed, do my permutational ANOVA on that, and I failed to reject the null. So now we're saying, okay, well, L is different from C, but L doesn't appear to be different from M. And then I can similarly do C versus M. So I've done all three comparisons. And I do that, and it also pops up as being significant. And so we can treat that as a post hoc. So based upon that, L and M don't appear to be different from one another, but both of them are different from C. And that's how we would treat it as a post hoc. It probably would be appropriate to do some kind of a correction, either a Hochberg correction or our friend the Holm correction for multiple comparisons on the same data. Um, in our case, I'm not going to bother doing that because I know that 1 times 10 to the negative fifth is going to still be significant if I had 3 multiple comparisons there. So I'm not going to really worry about it, but if we were to do it, we could do it. One thing to remind yourself of when you're doing permutation hypothesis testing and thinking carefully about how to interpret the p-value, especially if you want to do corrections, is that this p-value, how small it can get, is contingent upon how many permutations I do, right? Because it's basically looking at that null distribution. So if I had only done 99 permutations, like let's go something really, really small, that is the smallest possible p-value I could possibly get. Likewise, I bet that if I go to 999 permutations, my p-value is going to be 0.001. It is. And so that whole 1 times 10 to the negative fifth, that's because that's the smallest possible p-value you could get. In other words, there was, depending on how the rounding happened, but I feel probably good that it's the case, that there was not one permutation where the null hypothesized f value exceeded our observed f value. So just kind of be aware of that when you feel compelled to do any kind of correction for multiple comparisons in the end, is that your p-value is going to be defined by your number of permutations. So I could make that p-value smaller if I just increased the number of permutations. Which is why generally when you report these kind of things, this is an example of where it's totally fine to just say p less than 0.05. You don't have to give the exact p-value because it's entirely arbitrary, right? 1 times 10 to the negative fifth is only because I did 99,999. I could add another another digit to that, and then I would end up with 1 times 10 to the negative sixth, which sounds like a really smaller p-value than 1 times 10 to the negative 5, but at the end of the day, they're telling the exact same story because the p-value is contingent upon the number of permutations. One quick thing. When I said that the more you increase the number of permutations, the smaller the p-value gets, that's true in this case because our groups were so different. Basically, none of our permutations came up with an f value that exceeded our observed f value. So it's not true that as you increase the number of permutations, you necessarily will always get a smaller p-value. So hypothetically, if our p-value were 0.02, increasing the number of permutations will give you maybe a more precise idea of what the p-value is, but the p-value will not decrease, because if the p-value were 0.02, let's say, that means on average 2% of our simulated F distributions or our permuted F statistics actually did exceed our observed. So I just wanted to make that clear so you weren't under the impression that you can always get a significant p-value just by increasing your permutations. That's not quite how it works. I was just talking about our particular example here where the differences are so extreme that it's very unlikely that we would have a permutation of the data that would actually give us an F statistic that exceeded our observed. So I just wanted to make that clear so you weren't under the false impression that you can always cook up a really good p-value by increasing your permutations. Okay, sorry. So an RDA can accept any type of predictors that a linear model can. So you could you could treat it as a multiple regression. You could have multiple predictors in the model, and you would be able to do hypothesis testing on each of those, for example. So we totally have the abilities to do that. Right. I'm gonna take a little pause, just a little pause. All right, one more thing I want to mention. As I promised, there is one other thing I wanted to mention, and that is when you do have multiple different predictors in a model. So again, an RDA kind of framework can accept anything that a traditional linear model can. So you can have a mixture of continuous and categorical predictors. But what we need to think about is how to do the hypothesis testing when we have those multiple factors that go in there. So what I'm going to do for the butterfly data is I'm going to include another piece of information that I've been keeping secret, which is brood information. So these populations of butterflies have two broods, an early and a late brood. And we might be interested in knowing whether or not there's a difference in the wing morphology between early and late season, because it wouldn't be unheard of in the world of insects for brood to matter. So I'm going to have this object here that I am going to call brood. And there it is. Hey, I disappeared. Oh man, nobody wants to play with Jim. What? Oh, I know what I need to do. I need to make it float on top. There, I'm floating. And so we would go ahead, and the first thing we would do, just like in an MANOVA, is we need to have that one important step where we make sure that there's no significant interaction. Because if there is a significant interaction, that means that we really shouldn't be interpreting the hypothesis testing on our main effects. So what I'm going to do is I'm going to set up an RDA where I have populations and brood in the model, but I'm going to include the interaction term. Okay, and again, I'm going to say scale equals true because I want to do it on the correlation matrix. So I'll go ahead and I'll run that. And now when it comes to the hypothesis testing part, let me delete this for just a second. So this is how we had set up our hypothesis testing before, and if I ran that, it'll chug through all the numbers, chug it, chug it, chug it, chug it, chug it, chug it. There it is, and it's significant. But that's for the whole model. That's telling us that there's something significant in the whole model, but it's not telling us where those significant things might be. So what we need to do is we need to do the hypothesis testing on each term in the model. And so recall that when we use the capital A MANOVA function from the CAR package to do a linear model, we're using the capital A MANOVA function because it is calculating it based upon the margin, right? It's the marginal sums of squares, so it's treating each term as though it were the last thing in the model. That way order doesn't matter. And that's the same thing that we're going to want to do here. And so we're going to say by equals margin. And there are a few other options that you can look into. If you do it by equals terms, it would do it sequentially, so then order would matter. And you can also do by equals axis so that you can see which axes, which constrained axes are significant. But the main one that we'll want to do for hypothesis testing is to simply do it by equals margin. And so I will go ahead and I will do it by equals margin. And the first thing it's going to do is it's going to take that highest order term, which is the interaction term. So here's pops by brood. And it's telling me that it's not significant. And it's stopping there because it's basically saying you don't have a significant interaction term in there. Why do you have it in the model in the first place? And so once we look at that and we're like, yes, I'm totally happy with eliminating that interaction term, then I will go to a simple additive model. So I'll go ahead and I'll set my seed again. I should probably do this. And I will go ahead and I will run the PERMANOVA now on that. And it's chugging through all 99,999. And there we have it. We can interpret the main effects because there's no significant interaction term. We have populations is still significant. And now brood appears to be potentially of some interest. And if we were to look at the summary of that, just to point out what it would look like. Go up to the topsy tops here. Okay. Now we have, what do we have? Did I do the right one? PC, butt wing, PC RDA, summary. Yes, there it is. And now we have 3 RDA axes because there's one slope associated with early and late brood, and there's 2 slopes associated with our 3 different populations. So now we have 3 RDA axes. We're explaining a little bit more of the total variation than we did last time. Now we're up to 35.6% of the variation. And we could, as before, we could interpret, what are the important components here for RDA1 and RDA2, as well as RDA3, if we wanted to. But RDA3 is really not explaining that much, right? RDA3 is explaining 1.1% of the variation, which I would argue is substantially less than 31% of the variation. So next we might want to plot that, and we can plot that as we did before. Okay, so here's our new plot. It looks really similar to our old plot because we didn't really add that much explanatory power by putting brood in there. But what I'm going to do is I'm going to add another kind of graphical parameter to this to make it easier to look at, because right now it's just the populations that are color-coded. It's not what brood you're in. And so I'm going to change the plotting characters. So traditionally I always say this PCH, plotting character, equals 19 because that's a solid circle. And what I'm going to do is I'm going to create a vector, much like I did for colors. I'm going to create a vector where I'm going to say it's going to start out, everybody's going to get a solid circle, and then those individuals that are in the early brood, I'm going to give it the value of 1. And the value of 1 is an open circle. And so now I will put those PCHs into my plotting function. And now the open circles are the early brood. The later, the solid circles are the later brood. And you can kind of eyeball that and say to yourself, it looks kind of like if RDA2 is doing anything, it's maybe starting to pull apart early versus later brood, but RDA1 is really still all about the difference between the populations. And so yeah, so again, you can put anything you want into these models. You just have to, treat it like you would a linear model. Think about the underlying assumptions and think about the hypothesis that you're interested in testing. And so that concludes our wonderful, brief, albeit spectacular introduction to redundancy analysis. Again, as I've mentioned before, this book is free from the library as an online ebook. And there is a really well done chapter in here that's called Canonical Ordination, which is basically a fancy way to say constrained ordination. And it has a really, really nice introduction and description of how to do a redundancy analysis, how to interpret it. If you have a lot of predictors, there's even a built-in step function so that you can do ordi step, which will do stepwise model selection if you have a lot of potential predictors. And so those of you out there that are going to be dealing with multivariate data, and this is especially true for those of you that are doing like microbiome stuff or population genomic stuff where you have these large multivariate matrices, redundancy analysis and their kin will probably be your friends. So I would definitely say check out this book. And if you also just did a tutorial RDA vegan, there's a couple of really nice tutorials out there, including a tutorial by Yuri Oksanen, who is the person that's kind of responsible, the overlord of the vegan package. He has a really nice tutorial on how to set these up and how to think about things. Granted, this was a really brief introduction to it because I know from personal experience, it really is at least a 6-week portion of the course if we were to really dig in deep. So this was just meant to be a general introduction to the idea, how it works, and how you might want to implement it for your own data. So that is our end of redundancy analysis. I hope that you enjoyed it and that it was as fulfilling for you as it was for me. Have a good day.